I completed these two dashboards. What I did first is I did a bit of research. I asked ChatGPT to create me a list of primary motif for each cell type. There are 7 cell types: the muscle, the skin, the brain, the white blood cells, pancreas, those cell types that you can see in the list. And after that, there's actually a secondary motif, which is just 1:1. You cannot actually do a selection. There's only one secondary motif that is paired with the primary. And then there's also, same as the tails, it's also paired with given the cell type. So it's actually paired with the cell type. So for each cell type, it has a recommended filler, mid filler, and tail filler. So once you make those selections, it will also tell you the recommended other sequence in each location. And then in the next dashboard, that is when you can actually still make modifications, because in the first dashboard you can only select the primary motif given what the cell type that was selected. And so the next dashboard, you can change the secondary motif, and then the mid filler and the tail filler. And then for the fillers, you can also enter a count, which means that you can further repeat the letter. So for example, if it's an S, then you enter 2, then it becomes SS. Okay, and then you have those for each location. There's a finalized pattern sequence. And then for the second dashboard, it will then start scoring the whether it's an acid, a base, whether it's highly soluble or it's hydrophobic. So what does this mean? The score is really like, you have to think about it. Like if you want it like brain, it has to be highly soluble. Do you want to introduce amino acids that are hydrophobic? That's up to you. Do you want it to be more basic, more acidic? So something like that. So yeah, it really just like, it also has like, if you're like, what should I do? Like, it also has a recommendation for like other like amino acids given the cell type. Like here, the brain, like which amino acids are usually found there. So, and then it's also which ones are not very common. It doesn't really mean like that it shouldn't be allowed, or this is the most highly suggested. It's just tells you like which one is most common. So then you can change the filler. So it's all, you're like adjusting it, so that it will produce a certain kind. Like overall, you're adjusting the pattern sequence, so that overall it will produce something that that you wanted. That if you want it to be more, those are just 3 tests. There are more. Definitely the next dashboards will have more configuration, so you can kind of make more changes. But those 3 are just the minimum viable biological test given the cell type that was selected. So this one will definitely be more than 70,000 unique unique combinations, because you can also adjust the length. So only the first 3 motifs in each location is kind of fixed, and I made that decision because we want to make sure that we have something that is actually familiar that is already being used in each cell type. And the first in the N terminal is usually begins with M, and some of these primary motifs are actually repeated in different cells. So like there's like 280 like possible, like, because there's like 7 cell types times 10 each location, 10 times 4, 7 times 10 times 4, but there's only actually like I think 100 something that are really unique primary motifs. So kind of, but the functions are different, even though for example, I don't know what it is, but maybe MKS or something like that, maybe it's some are actually repeated like 17 times, not just in the first N terminal, it can be used in other locations, but they all have different purposes. So you can read the description and see what kind of function, like what they're actually doing for each when it is located in different locations or in different cell types. So kind of have like so it's not just like, so can have some primary motifs, which is kind of important when making the patterns, that you kind of have like even like just a basic syllable, and then you can add fillers and secondary motifs. So that's what I have right now. So it's kind of my first attempt to create regenerate my own protein sequence, that we can actually solve for. We can do tests, and then kind of think about then what we want it to be, to kind of have a report and see if that fits into our goal. If and we have more tests, and then we can make adjustments as necessary.